Okay, hi everyone. Welcome to our series on cerebellar research. Today I have as our guest Sang-Jung Kim. Sang-Jung was trained as a physician at Seoul National University and received his MD in 1990. Then he stayed on to complete a PhD in 1996. He was a postdoctoral fellow at Johns Hopkins with David Linden and Paul Worley. And during this work, he discovered that in addition to typical mechanisms of synaptic plasticity, LTP and LTD, there are also other mechanisms that with which neurons can store memories. He then returned to Seoul National University, where he's now the chair of the Department of Biomedical Sciences and the director of the Memory Network Medical Research Center. He's won a number of awards, including when he was young. In 1999, he received a Young Physiologist Award from the Korean Physiological Society. And then more recently, in 2021, he received the Joseph Jin Chang Research Award at the 2021 Korean National Neuroscience Society. He studies cellular and molecular mechanisms of long-term memory, focusing on the cerebellum. And it's a pleasure to have you with us. Look forward to your talk, Sang-Jung.
Uh, Thank you very much for your kind introduction and for inviting me to this wonderful seminar series. I'm so delighted to share the stories of my brilliant students, Hyungun and Changyeon and Jae-Wen Dongchul, on regarding on memory consolidation in the cerebellum. Allow me begin with my neural tree. And as you introduced, I did a postdoc at Johns Hopkins at David Linden and Paul Worley lab, and I learned cerebellum at Johns Hopkins. David traveled all the way to Seoul to celebrate the opening ceremony of my Memory Network Memory Medical Research Center in SNU. I deeply appreciate my mentors, Professor John Kim and David Linden and Paul Worley in Johns Hopkins. Okay, to me, cerebellum looks like a conductor of a symphony orchestrating brain function. Purkinje cell in the cerebellar cortex is kind of a very talkative. It talks a lot, and I don't know whether you can hear the sound. This capture of extracellular recording sounds very talkative. It's brook, and so it shows beautiful simple spike and complex spike. So as a neurophysiologist, this talking really intriguing and then really triggers my curiosity. How does Purkinje cell maintain this firing, and why PC fires a lot? And during the Purkinje cell firing, the calcium influx is unavoidable. So Purkinje cell need to manage this calcium influx to sustain Purkinje cell firing. For this end, Purkinje cell is equipped with very effective calcium handling strategy, which is abundant calcium buffering molecules like carbamide and parvalbumin. Also, it has calcium store. And STIM1 is an calcium sensor which binds to calcium pump in the membrane of calcium store. Changyeon Liu, currently a postdoc at MIT, decided to hypothesize that STIM1 may contribute to calcium handling in Purkinje cell and overall contributing to Purkinje cell firing. And to this end, he generated Purkinje cell specific STIM1 knockout mice. And he crossed flox STIM1 and PCPT3 mice line. And this immunohistochemistry revealed that the absence of expression of STIM1 in the Purkinje cell soma and the dendrite. This floxed STIM1 line is general gift from Paul Wally lab. And Changyan measured the calcium concentration in the Purkinje cell soma and the membrane depolarization, and also Purkinje cell firing induced increase in the intracellular calcium concentration. And the knockout of Purkinje, the Purkinje specific STIM1 knockout delayed cytosolic calcium clearance compared to wild-type mice. From now on, the blue represent wild-type and the red represent knockout mice. So this delay in cytosolic calcium clearance may affect, calcium-dependent membrane conductors, and then that eventually affect the Purkinje cell firing. And we measured Purkinje cell firing with serotoxic mode and found that the Purkinje cell specific STIM1 knockout reduced intrinsic excitability of the Purkinje cell compared to wild type. So to understand why Purkinje cell fires even with this enormous energy consumption, we investigated the impact of Purkinje cell firing deficit in STIM1 knockout on cerebellum specific behavior, which is vestibular ocular reflex. So you can see the turntable and mouse on the turntable, and then you can see the screen of the equipment. This vestibular ocular reflex system was transferred from Dulac lab. I do appreciate her contribution to this story. And the VUR learns adapt in the training, during the training. For example, if the turntable and screen turns in opposite direction, the movement of eyeball gain increases. On the other hand, if the turntable and screen rotate in the same direction, the eyeball movement gain goes down, gain decreases. And we tested this visual gain, for example, this gain down learning, 2 hours of price. These Purkinje cell specific STIM1 knockout mice, the VUR gain learning was intact. So, this is gain down, and there's no difference in the the memory of the VUR learning. But when we come back to this mice, next day, these mice forget the previous day memory. So you can see the memory gain goes back to a baseline at 24 hours. So this STIM1 knockout mice shows kind of phase-specific deficit in the memory consolidation compared to wild type. And also, when we did the gain-up training like this also gain-up learning was okay, but the gain-up consolidation showed deficit in STIM1 Purkinje cell knockout mice. So we found that STIM1 knockout in the Purkinje cell induced memory consolidation deficit, but the basal firing deficit, which I have shown you, may not explain this very specific deficit. So to understand the underlying plasticity mechanism, we decided to study temporal correlation between the neuroplasticity and memory. Hyung-geun, Shim, and Dong-chol did this work together. And so we know that there are two types of plasticity. And for example, parallel fiber burst induction induces synaptic LTP and intrinsic plasticity potentiation together, which are called LTP-IE. And also our lab also reported that different induction pattern induces synaptic LTD and then LTD-IE together. So this is data from AcuSlice in vitro induction protocol. So our lab decided to investigate training-induced plasticity during VUR learning and memory consolidation. So we did, in this case, gain up training, and then this is the gain. Gain is increased by this training of 30 minutes, and then if the memory consolidation was intact in wild-type animal. And we sacrificed this animal at every point of this behavioral measurement, and we investigated the intrinsic excitability of Purkinje cell. And so this gain of training induced LTDI, which means the depression of intrinsic excitability at 1 hour, and it returns back to baseline after 24 hours. So we did this recording from the flocculus, which is reported to be the site of the VOR memory storage. And then we tested knockout mice, and as I shown you before, knockout mice learns okay, but the memory consolidation wasn't okay. So memory gain returns to the baseline, and then we checked the intrinsic plasticity, and then we found that at 1 hour, we could not see any change in intrinsic plasticity, LTD-IE, and 4 hour and for 24 hour. So the STIM-on knockout mice showed memory consolidation and LTD-IE deficit after gain-up training. And how about synaptic plasticity which is induced by VOR training? So in contrast to intrinsic plasticity, the synaptic plasticity was intact in both wild type and STIM-on knockout mice. So as you can see here, after VOR training, gain-up training, after 1 hour, there was a synaptic depression both in wild type and knockout, and it returns to the sham level after 24 hours. So then we tested that whether Purkinje cell instruct VN neurons for memory transfer. It is so we tested vestibular nucleus, Moshe fiber to vestibular nucleus synaptic plasticity, and also vestibular nucleus intrinsic plasticity. Due to the time limit, I want to only show the intrinsic plasticity of vestibular nucleus. So the training, after the train, and at each time point after training, we measured the intrinsic plasticity from the vestibular nucleus in ex vivo or whole cell recording mode. So in wild type mice, after 1 hour, there is a tendency of increasing intrinsic excitability, and 4-hour and 24-hour it keep increased. But in STIMUL in knockout mice, this LTPI training induced LTPI in VN neuron showed deficit. It did not show any intrinsic plasticity in VN. So I'd like to summarize this part, and then I'm showing you that this is a behavior data. So gain up, and then it is consolidated after 24 hours in wild type, and I've shown you that this VUL training induced synaptic plasticity in Purkinje cell, and also intrinsic plasticity in Purkinje cell. And also subsequently, there is a synaptic and intrinsic plasticity in vestibular nucleus, which you can see here. So this is wild type, but in the knockout, there's a deficit in intrinsic plasticity, and then the synaptic and intrinsic plasticity deficit of VN follows this intrinsic plasticity deficit of Purkinje cell. Also, this deficit has a correlation with the memory consolidation of the VUL training. So we can say that deficit in Purkinje cell intrinsic plasticity correlate with deficit in memory consolidation and vestibular nucleus plasticity. So then this temporal correlation does not prove the causality between intrinsic plasticity and subsequent memory consolidation and VN plasticity. May not. So Jeu, my student, decided to test the causality between Purkinje cell intrinsic plasticity and memory consolidation. To test this causality, we developed optogenetic manipulation of Purkinje cell intrinsic excitability using channel adoption, which is a step function option, SSFO, which means we can turn on this option with blue light and turn off this option with yellow light. And we targeted flocculus, and then we expressed channel adoption, and also control EYFP. And we made an acute slice and measure the intrinsic excitability in each condition. So in control, this laser turn on and turn off did not change the intrinsic excitability of Purkinje cell. On the contrary, step function option, this activation of step function option increased the Purkinje cell firing, and then the step function option turn off decreased the Purkinje cell firing here. So you can see this step function SSFO increased the Purkinje cell firing. So then we applied this optogenetic manipulation during consolidation phase. So here you can see that we trained these mice. In this case, we used actually optokinetic reflex training, and we trained 15 minutes, and then so we compared between EYFP control and then the SSF for mice. So this is OKR training configuration, and after 15-minute learning, there's an increase in gain in the eye movement, and here you can see, and then there's a memory consolidation in after 24 hours. Compared to this EYF control, this optogenetic manipulation during consolidation phase period with SSF4, the memory consolidation will show deficit. So just post training, there is a comparable memory. I mean the gain increase after OKR training, but this manipulation, SS4 manipulation, induced gain, OKR gain back to baseline. So here you can see the consolidation percentage. So in YF control, you can see 100% consolidation. But in SSF4 condition, you can see a deficit in memory consolidation. So then we investigate. So you may remember that we are training the intrinsic plasticity potentiation. I mean, depression was transient, like between 1 hour and 4 hours. So we tested whether there is any critical period for the intrinsic plasticity for the memory consolidation. So this is data manipulating from a whole 24 hours, which I have shown you last slide. So we can see there's a memory consolidation deficit in 24-hour optogenetic manipulation, and in this right side we change the duration of optogenetic manipulation from 90 minutes after training to the 24 hours. So we spared this 90 minutes period spare this period without any optogenetic manipulation. So you can see that this 90 minute to 24-hour manipulation did not affected memory consolidation, both in EYP and step function opsin condition. So it means there is a early phase period is critical for the memory. I mean the Purkinje intrinsic excitability contribution, and we tested this finding again, applying this optogenetic manipulation only from 0 minute to the 90 minute post training. So as you can see here, this EYP didn't affect memory consolidation, but from this step function option, SSF condition, the 0 to 90 minute optogenetic manipulation of Purkinje cell firing affected the memory consolidation like this. So there in EYP, there's a 100% consolidation, but in SSF for manipulation during 0 to 90 minutes post training, induced deficit in memory consolidation. And Zhou confirmed that the Purkinje cell intrinsic excitability dynamics during the critical period window, as I have shown you, from 0 to 9 minutes. And he did the same ex vivo measurement of Purkinje cell excitability as we did with VOR training. So after OKR training, in this case, OKR training, he sacrificed the animal and sliced the flocculus, and then patched the floccular Purkinje cell here and 0 minute after training, and then also he did 90 minute. After 90 minutes, he did also slice preparation. So you can see here that compared to control intrinsic excitability, the 0 minute Purkinje cell showed decrease in intrinsic excitability. You can see here, and then this decreased intrinsic excitability returns back to control after 90 minutes of training. So the intrinsic excitability depressed and then returns to baseline in 90 minutes. So you can see here the parameters of real base current and AP threshold and amplitude and this kind of data here. And how about the VN plasticity in this optogenetic manipulation configuration? So we did same OKR training, and we prepared, in this case, vestibular brainstem slice, including vestibular nucleus. And we also injected rhodopsin or EYP in Purkinje cell, and this Purkinje cell axon goes to vestibular nucleus, and we can see this. We can see that right here. You can see that the Purkinje cell terminal projects to follicular targeting vestibular nucleus, and we can identify that vestibular nucleus neuron which received the terminal axon terminal of the Purkinje cell, and then we patched this follicular targeting vestibular nucleus, and then we measured intrinsic excitability of vestibular nucleus. So as you can see here, this is EY control, and this is SSF condition. And then in control, so we measured the intrinsic excitability of vestibular nucleus pre-training and after training, and after 24 hours training. So compared to before training, after training, you can see increase in membrane excitability in vestibular nucleus neuron in EY control, and in step function option condition, you can see that this optogenetic manipulation for 24 hours of Purkinje cell intrinsic excitability disrupted this kind of long-term potentiation of intrinsic excitability of vesicular nucleus. So you cannot see any difference in intrinsic excitability between pre and post 24-hour training conditions. Okay, then as a last part, so we were curious. We are curious about simple spike contribution to memory, cerebral memory, and then which is really spontaneous firing. But also there's another spike, complex spike also, regularly in Parkinson cell shows like every 1 second Parkinson cell it fires complex spike also. So we questioned about complex spike also contributing memory consolidation or memory acquisition or memory retrieval. Or so we asked this kind of questions, and Jeo then decided to investigate complex spike contribution to the motor memory acquisition and consolidation and retrieval. So you can see as you can see here, the Klein-Fiber Parkinson cell fires complex spike driven by intrinsic excited input from Klein-Fiber, and then so he expressed halo rhodopsin in the olivary nucleus, which projects Klein-Fiber to the Purkinje cell, and he injected here in the olivary nucleus, and then we can see the expression of halo rhodopsin in the Klein-Fiber in the cerebellar cortex. And you can see here is a molecular layer, and you can see the Klein-Fiber fibers which express halo rhodopsin, EYFP, or this case is a control EGFP. And we made a flocculus slice and tested whether this halo rhodopsin suppress Klein-Fiber complex spike. So we made a whole cell recording here, and then we stimulated Klein-Fiber, and then we measured the Klein-Fiber evoked EPSC here, and then this control EGF mice optogenetic stimulation did not change this Klein-Fiber induced EPSC, but on contrary, the halo rhodopsin expressed in Purkinje cell showed that optogenetic manipulation inhibited this Klein-Fiber evoked EPSC here. So you can see the suppression is pretty complete in this condition. And then we optogenetically manipulated Klein-Fiber transmission at the specific phase of the memory process. In this case, we interrupted the Klein-Fiber transmission during memory acquisition phase. So as you can see here, we applied laser stimulation during memory acquisition period, during training period. So we trained this animal like 5-10 minutes training. So here you can see the optogenetic inhibition of Klein-Fiber transmission during acquisition. And in GFP control, you can see the optogenetics. I mean, the laser stimulation did not affect the learning process, but this halo absorption activation in the complex climbing fiber um, uh, interrupted memory acquisition, as you can see here in the gain. So there is a compared to GFP condition, halo absorption condition, there is a interruption in the memory acquisition. So you can see the row trace here, and then compared to GFP, halo absorption mice did not learn pretty well. And how about the memory consolidation phase? And we applied this optogenetic manipulation during consolidation phase, as you show in red color, yellow color. So in this case, we applied like a 30 minutes case, and this 30 minutes of optogenetic suppression of client fiber did not show any change in memory consolidation percentage here. But maybe this 30 minutes was not long enough because we have shown you that maybe 90 minutes after training is critical for the memory consolidation instructed from Purkinje cell. So we increased the duration of optogenetic manipulation to 6 hours. And this 6 hours manipulation also did not affect memory consolidation. Also, we tested the optogenetic manipulation during memory retrieval. So after 24 hours, we applied optogenetic manipulation, but memory retrieval was not affected by this manipulation. Let me summarize my presentation today. So I've shown you that Purkinje-specific Steeman knockout mice showed memory consolidation deficit in VOR learning and memory. And we also showed that VOR training induced intrinsic plasticity in Purkinje cells. But in the Steeman knockout mice, this intrinsic plasticity was impaired, but synaptic plasticity was intact. And to test causality, we optogenetically manipulated Purkinje cell intrinsic plasticity during memory consolidation period, and then manipulation impaired memory consolidation and also VN plasticity. And finally, optogenetic manipulation of client fiber transmission interrupted memory consolidation, but not memory consolidation. So I'd like to suggest that intrinsic plasticity in Purkinje cell is required for the change in downstream plasticity in VN, contributing to the system's memory consolidation of motor memory. And here's a one fine day in Seoul. It's beautiful skies, and this is like a Teacher's Day. So we got me and Yong-Sung Lee, who's running this neurophysiology lab together. Was so happy to be together with all the very motivated students. thank you. And also, I'd like to express my really uh, uh, thankfulness to Dr. Paul Wally and David Linden. Thank you for your understanding. I mean, thank you for your attention. Thank you very much.
Well, thank you very much. That was just wonderful. Please, if you have questions, just raise your hand, and I'd be happy to uh, give you an opportunity to ask Sangjong your questions. Um, the change is an uh, um, intrinsic plasticity. Can I interpret in the Purkinje cells? Can I interpret that as baseline firing rates that change after the adaptation phase and then return to baseline to within 24 hours?
That's right. That's right. Yes. It's kind of engaging during the critical period and returns back to baseline. That's what we have seen.
Um, and is there any change in the Purkinje cell vestibular nuclear synapse? You showed that there was a change in the mossy fiber um, vestibular nuclear synapse. What about the P cell synapse?
Yeah, we haven't recorded that synapse. You are wondering the synapse between Purkinje cell and the vestibular nucleus plasticity. No, I don't have any data about that. Mhm.
Yeah, yeah. Yeah, that's it's really it's it's a it's an amazing I feel it's an amazing result. The changes that take place in the synapses that then somehow persist in the activity of the cell during the offline period that then becomes important for downstream plasticity in the vestibular nucleus. Anyway, I found it amazing. I don't know if you have any questions for Sang Jong. A follow-up, I guess, but the climbing fiber activity you showed was critical for acquisition, but then its suppression during the consolidation period did not have any effects on maintenance of that the downstream effects. So, what's the relationship between climbing fiber activity and intrinsic plasticity?
Um, yeah, I mean, uh, in acute slice preparation, so uh, it's like synaptic plasticity. If I pair parallel fiber and client fiber together, the client fiber contribution is the polarity of plasticity goes to depression. If I just induce plasticity by stimulating parallel fiber only without client fiber activity, the synaptic and intrinsic plasticity polarity is to the direction of potentiation. So that's kind of a acute slice finding. So, but in vivo configuration, we haven't checked. So we need to do kind of ex vivo experiment, and then before and after manipulating client fiber activity. So so we haven't done that experiment yet. So but that's really uh, exciting experiment I would be. Yeah.
Systems consolidation was first hypothesized for the hippocampus and the cerebral cortex. Um, what mechanisms do you think may be in common between what you've shown here with the cerebellar cortex and nucleus and systems consolidation and, like episodic memory that's associated with the hippocampus?
Yeah. Um, so, uh, maybe. So, I mean, it's a very critical question. I mean, so for example, in the hippocampal, I mean, the episodic memory or that kind of hippocampus related memory wise, people has recorded, the offline activity like, sharp wave ripple or something like that. So
or during sleep?
yeah, during sleep, yeah. So that's a pretty good evidence or a possible candidate to support the kind of offline teaching. I would say instruction signal from hippocampus to final destination brain areas. So comparing to that hippocampal field, we need data from several area. The kind of offline activity is really engaging. We don't know any kind of a brain wave or kind of instruction really really should be measured and then tested. So that's kind of a further experiment I'm interested in.
Right. Very good.
Can I ask a question,
Yeah,
Reza?
David, go ahead.
In in humans, we're quite we're quite foveate. When we do these up and down VOR adaptation experiments, especially with up, we often see the subject incorporating saccadic eye movements to catch up to the target, if you will, and that becomes automatic. Also a learned thing. So my question is, in your model, do you ever see any change in the quick phases or the saccadic mechanism trying to also incorporate some correction for the retinal image slip?
Oh, yeah. I mean, I haven't checked. Oh, yeah. So my quick answer is we haven't looked at that much carefully. So yeah, I will come back to our data and then yeah, I will analyze that. I mean, the
Send
Sakari.
send me an email.
Yeah. Thanks very much.
Okay, thank you.
Wonderful. Sangjung, it's been a pleasure to watch your work. It was really wonderful. I learned so much from it, and I hope everybody enjoyed it as well. Thank you so much for your time, and have a wonderful evening.
Yeah, thank you very much. I mean, uh, thank you very much for participating so early in the Eastern time. Now I'm going to bed. Thank you very
Okay.
much.
Good night. Bye bye.
Yeah. Have a good day.